So chapter 10 focuses on meiosis and sexual reproduction. Chapter 9 we did mitosis. You should also be working in your Connect and keeping those assignments on track as well. I'll try to extend some of the due dates so you have a little more time to complete those in the Connect portion of your class. So the importance of meiosis or meiosis introduces an enormous amount of diversity. And remember that we use this with sex cells instead of those somatic cells that we used with in mitosis. There are more than 70 trillion different types and possibilities when we mate two individuals. Males and females differ in the way they form gametes. And so the word gametes, when we're talking about that, we are going to be talking about sperm production in males and egg production in females. In males, the sperm production begins at puberty, but in the female, the process of producing eggs starts before birth and ends at menopause. So an overview here, remember it's only for sexual reproduction. It's a special type of cell division, and mitosis we ended with two daughter cells, but with meiosis we're going to end with the 4 daughter cells. So they do have the same phases, but meiosis, remember, is going to undergo those twice so that we get them really individualized and so that they are not the same exact combinations as their parents are, so we do not produce clones. And we'll do some special things here we'll talk about in a minute called crossing over and how that occurs and how that makes us different and who we are today. So the chromosomes are replicated in the S phase of interphase. Remember, interphase, we spent 90% of the cell cycle in that. And remember from mitosis that parents are diploid, which we said was 2N, and haploid, which was N when you go to write these. And the gametes fuse in fertilization to form a zygote. Your zygote is going to be the baby, if you will. And we say that they are diploid or 2N because they have the correct number of chromosomes when they come together. Now, unfortunately, we do have events where meiosis will go wrong, where we can have mutations. So gametes can contain the wrong number of chromosomes. They can have too little, they can have too many, and those can cause mutations in your offspring. And I'll talk about some of those at the end of this chapter here. Remember that humans have 23 different types of chromosomes, and they are the same length. And if they are the same length, remember they are called homologous chromosomes. I think we talked about that last time in our lecture, last time in class. Their centromere, remember that was the center of our X, if you will, are positioned in the same place. And one came from the father and the other from the mother. And so we put them together with the centromere in the middle. And when we stain them in labs, they're going to show similar banding patterns. So here's an example of that where they're stained and looked at under a powerful microscope. And you can see these here and how they are stained, and they do look the same. However, when we have mutations, they will look different when we are examining them in the lab. And so here we have one chromosome from the father, and we have one chromosome from the mother. And so we can put them together, remember, with the centromere in the middle, and if they're the same length, we call them homologous chromosomes. So just like in mitosis, these phases are pretty similar, but since they undergo 2 nuclear divisions, the first process we call is the first division, and then we're going to have the second division. And underneath each of these divisions, so meiosis 1, we're still going to have those prophase, metaphase, anaphase, telophase, but after those words we will put 1. So prophase 1, metaphase 1, and then after we complete that, we got our 2 daughter cells. We will start the second process of meiosis, which will undergo prophase, metaphase, anaphase, and telophase again, but we will call them, we'll put the Roman numeral 2 after those. So prophase 2 now, metaphase 2 now, and so on and so forth. and we will end up, again, remember, with 4 daughter cells. In mitosis, we said that the 2 cells had identical DNA inside them, but remember, for meiosis, they are going to be different in each one. So they are not identical to each other. So an overview here, we have our first phase, meiosis 1, where homolog pairs synapse and then separate. So synapse is in this previous one here, where they're going to pair up. And this might be a question on your worksheet. What is synapsis? And it's going to be where chromosomes pair up. And so we can see that on this next slide where they are pairing. So we're going to have one of those X's from the mom, one of those X's from the dad, and so on and so forth over here, where they're going to pair up. And we have larger ones, we have these smaller ones, and they will pair up, and then they will start to separate out. And here, as we move on to the second phase of meiosis, you can see them breaking down into haploid daughter cells. Haploid meaning we just have one strand, diploid meaning we have 2 strands together, if that helps you as well. And so our 4 haploid daughter cells are all not identical to where we started, and so this depicts that pretty well. And once again, we've got the 4 new daughter cells there. Alright, so meiosis brings about genetic variation in 2 ways, and so this is another homework question, and one of those is going to be crossing over, like I said earlier. And it's where the homologous chromosomes are going to do something called independent assortment, where they're going to switch genetic information around so that we don't have the exact same offspring. So crossing over involves an exchange of genetic material between non-sister chromatids during meiosis 1, and I'll put a video in Moodle here over an example of crossing over. There's also a video here in this PowerPoint that you can watch, and you can watch it happen. And so you can just click play on this and it will walk you through it. It does have audio, so make sure you turn your speakers up whenever you're listening to this. But basically, what we're going to do is take some genetic information here, and we're going to just switch it to another chromosome. So you can see how the blue and the red kind of switching around here in different places, making new daughter chromosomes. And so here we have a solid red and blue bands. Now we have spotty red and blue bands, and now they have switched information around a lot. And so we call this crossing over when we do so. So next up is the independent assortment. This is just another way we can get genetic variation. When a parent cell has 3 pairs of homologous chromosomes, there can be 8 possible chromosome alignments at the metaphase plate due to independent assortment. So this is just going to be a random combination that's going to be thrown in there so that we have a lot of genetic variation. When we do have crossing over occur in meiosis, you can see that number, and I won't try and say this number, but all of that number is genetically different zygotes that are possible. And crossing over doesn't occur just one time, the switching of the chromosome information. It can happen several times in each chromosome as well. What is the significance? Well, the significance is that we're genetically different, so that we don't produce identical offspring. We want to have only our best genes survive, if you will, and it's going to give a better chance of survival for our offspring. So an example, if the temperature rises due to climate change, an animal with less fur or reduced body fat would have an advantage. And so this is letting adaptation happen as we go through time. And so I'll let you read on your own the phases of meiosis. They're the same as mitosis, so you can read through these, but it's going to happen twice now instead of just the once. And so you can read through these on your own. And remember, cytokinesis is the splitting of cytoplasm. We talked about that in class. Here is meiosis 2. So now we're just going to put that Roman numeral 2. So it's going to do all those phases again, where we are going to contain 4 haploid cells. There's a video as well in here in the PowerPoint. You can watch this and click the play, and it'll show you some features of meiosis there. I'll also put some more YouTube videos in here. And so again, I'll let you read through all these phases on your own. My most important thing just to know about this chapter is knowing the difference of meiosis versus mitosis. These are going to be the things that will be on your exam. These are going to be on your homework pages. This is just going to be the most important aspect of this PowerPoint.